children, yesterday we have done the structure of a ovule. In this section, we will discuss about the various parts of ovule in more details. So the very first structure is integument. We know that integument is the outermost layers of the ovule. Sometimes the integument can be 1, it can be 2, even 3, but not more than 3 as I have told you yesterday. And sometimes the ovule is even ategmic, that means it is without an integument. Integument in the later stages, when the ovule develops in the seed coat, becomes a seed. The integument forms the seed coat. On the basis of the number of integuments, it can be of various types. Number 1 is unitegmic, that the name suggests us that uni means one integument is there. Bitegmic, 2. Tritegmic, 3. Ategmic, without any integument. We will discuss with the examples then. Unitegmic examples are all the gymnosperms, and then we have the higher dicots. Bitegmic example is Malvaceae family. Malvaceae family has some members like China rose, ladyfinger. Crucifery, one of the member is mustard. Liliaceae is the example of a tritegmic. Liliaceae, in the Liliaceae family we have some members like lily. The scientific name of lily is lilium. Then we have onion, that is Allium cepa. Onion, that is Allium cepa. Then ategmic, that is without any integument, that is Santalum, and we have Loranthus. So these are some of the examples on the basis of number of integuments present in the ovule. The second structure, a very important structure, is nucellus. Nucellus is just inside the integument. It is the outer part of the embryo sac. Yesterday we have discussed about the diagram. Nucellus is the one which provides nourishment to the growing embryo. This provides nourishment to the growing embryo sac. That means it is used up while the embryo sac is developed. But in some cases, the nucellus still remains persistent throughout in the ovule. In that case, the persistent nucellus is called perisperm. This is a very important point. Perisperm is observed in beetroot, even pepper. This is called as remnant. Remnant means remains of nucellus still prevails inside the ovule, then that is termed as perisperm. Examples are beetroot and pepper. Do remember these examples. These are very important. Then nucellus is a diploid structure. We know that integument is also a diploid structure, and this is actually a sporogenous tissue which is present outside the embryo sac. It helps the embryo sac development, but I told you in many cases it remains there. It remains as a remnant, nucellus, which is called as perisperm. Next, we move on to the next section, that is the embryo sac. We know embryo sac has synergids, and we know the egg. One egg along with the 2 synergids is called as egg apparatus. The egg is also called as oosphere. In synergids, synergids, we have one of the very important finger-like projection structure which is called as a filiform apparatus. We have to remember that filiform apparatus helps in guiding the pollen tube into the embryo sac. And second, when the pollen tube enters through this, that is the filiform apparatus, here the pollen tube bursts, releasing the two of the male gametes here. One of the male gamete will move to the egg, another will move towards the polar nuclei. That we will discuss in the later sections. And therefore, when it bursts here, it also helps in absorbing the shock. The shock is absorbed by the filiform apparatus. We know that we have two synergids, therefore two filiform apparatus. One thing more I want to tell here is that this synergid, this is a structure of a synergid. You can observe here that it is a pear shaped. The pear shape is called as pyriform. Pyriform. That means the synergid is pyriform. One more thing, we have that embryo sac has two synergids and the entry of the pollen tube occurs through only one of the synergid. When the pollen tube enters through one of the synergid with the help of a filiform apparatus and it bursts, the fertilization occurs. In the rest case, what happens? The second synergid from which the entry has not taken place will degenerate. In the same way, the antipodals which are present above the embryo sac, they also degenerate. They are just there to provide nourishment and they, in the later stages, they degenerate. There is one chalaza end. Chalaza end is a very, very active part of an ovule. This is the part from where the integuments arise. Therefore, it is called to be an active part of an ovule. Clear? Micropyle end is the small opening. We have discussed yesterday in the structure of an ovule that micropyle was there. This micropyle is the opening through which the pollen tube enters into the ovule. When the ovule develops into a seed, you must have seen the Rajma seed. There is a small opening there. That is a micropyle. We know the Rajma kept in our kitchen containers, they are at the dormant period. That means they are still viable. That means when we just take it out them and we give the favorable conditions, they can sprout into a plant. They are still viable. That is called as a dormant period. That small opening which still remains there, that is called as a micropyle through which the gaseous exchange takes place. So that means that seed remains live for that duration. We have discussed about the antipodals, micropyle, chalaza. Chalaza we have discussed with the synergids. I have told you the functions of the filiform apparatus also. We have discussed the embryo sac completely. We have discussed about the egg apparatus also. We have to remember this. I am repeating again. Egg apparatus is what? It is two synergids plus one egg. Egg is also called as a oosphere. We have discussed about the integuments and we have discussed about the nucellus. In the next section, we will discuss how this embryo sac develops. That means how the megaspore is formed. In the same manner, like microspore develops, the megaspore also develops, and that is called as megasporogenesis. In the next video, I'll show you that. Thank you.